glucose 6-phosphate dehydrogenase deficiency is a red cell enzymopathies. What are enzymopathies? These are pathologies of the enzyme. That is, a disease state that result from when the enzymes is either deficient or is dysfunctional. Enzymopathies refers to a group of disorders that arise from the dysfunction or deficiency of specific enzymes. Enzymes are very necessary for metabolic processes in the body, processes that lead to energy production, conversion of lipids to energy, conversion of glucose to energy, conversion of amino acids to other substances require the actions of enzymes. So these enzymes are necessary. But when they have issues that either they are not being produced in enough quantity, or they are not being produced at all, or they can no longer perform their catalytic function, then we can say that's enzymopathies. What are enzymes? Enzymes are biological catalysts that facilitate biochemical reactions in the body, and therefore, their proper function is crucial for maintaining metabolic processes. When enzymes do not function correctly, for reasons like genetic mutation, environmental factors influencing it, and other reasons, they can lead to a range of metabolic abnormalities and diseases or disorders. And all of these, when they lead to a particular disease condition or disorder, we can now call that group of disorders enzymopathic diseases or enzymopathic pathologies. Let's look at classification of enzymopathies. Enzymopathies can be classified in various ways. One, by the type of metabolic pathway that is affected. Based on this classification, using metabolic pathway that's affected, we are trying to look at what chemical reaction or biochemical reaction is affected. Let's say we are looking at a pathology, enzymopathology that affects the process of conversion of glucose into energy. There are processes that the body undergoes in order to reach to the production of that ATP energy required. So if an enzyme that is responsible for that process is deficient or dysfunctional, that leads to an enzymopathy. As such, we classify such enzymopathy based on the metabolic pathway that is what affected. Another way of classification is by inheritance pattern. Some of these enzymopathies are genetic, that is, the parents can give it to the offsprings. And we try to look at the pattern of inheritance. Is it dominant? Is it recessive? Or is it autosomal? Or is it sex-linked? Some of them are transmitted or transferred through sex-link, that is, only on the sex chromosome, X and Y. That's where we find the condition. Some are also transferred to their offspring through autosomes, which are chromosomes that are not sex-linked or that are not sex genes. Another way of classification is by enzyme function. We try to classify enzymopathies by enzyme function, what the enzyme does. Now let's look at enzymopathies being classified based on metabolic pathway affected. One is carbohydrate metabolism abnormalities. Under this, we try to look at disorders such as phenylketonuria, which is a disorder that is that results from deficiency in the enzyme that helps in metabolizing carbohydrate into energy. Another example is lipid metabolism abnormalities, which is a disorder that results from abnormalities of the enzyme necessary for the conversion of energy from fats. Another example is amino acid metabolism abnormalities, which is an enzymopathy that results due to the deficiency or dysfunction of the enzyme necessary for the conversion or proper metabolism of branched chain amino acids. So these are categories of enzymopathies being classified based on metabolic pathway affected. Another way of classifying enzymopathies is by inheritance pattern. I'll be able to let you know that an autosome is a non-sex chromosome in humans. And an autosome gene is one that is located on the autosome. And the autosome is not a sex chromosome. So therefore, inheritance of an autosome is independent of whether you are a male or a female. So under these categories of enzymopathies, we have the autosomal recessive. In this type of classification, these enzymopathies occurs only if the new individual inherited 2 copies of the altered gene from both parents. So you cannot come down with this condition if you only inherited 1. But once you inherited the abnormal gene that is having problem in the production of that particular enzyme or for alteration of any kind, you cannot come down with the disease condition. But once you inherited from your father and your mother a defected or altered gene from parents, then you come down with that. And that's known as autosomal recessive. But for autosomal dominant, you only need 1 copy of the gene, is it from either your parents or either from father and the mother. And once you have it, you must come down with that condition. Example is familial hypercholesterolemia, which is an enzymopathy that affects the cholesterol. Whereas cystic fibrosis requires you to have the altered gene in the autosome from both parents before that child can have cystic fibrosis. Another classification of enzymopathy based on pattern of inheritance is sex-linked. Like we said, this type of disorders are only linked to genes on the X chromosome. That is, we have 2 sets of sex chromosomes. One is XX female and XY male. So when the X linked, that means once you have the altered gene in the sex linked, in the sex related gene, that is either X or Y, your chances of coming down or not coming down is there. So for sex linked, the alteration is already on the X chromosome. So this is another pattern of categorizing enzymopathy. Another way of categorizing enzymopathy is by enzyme function deficiency disorder. This is based on the function of that particular enzyme. You can classify enzymopathies based on the function that particular enzyme does. We can do that based on deficiency disorders. This is when the enzyme is completely or partially deficient. That is, it is not available. That enzyme is not available in that particular process. But ideally, it's supposed to be there. But for whatever reason, the body is unable to produce it. As such, leads to accumulation of substrates and a lack of products. So because that enzyme is not there to catalyze the process, there will be lack of that particular function or lack of production of that necessary hormone or whatever that is necessary for that part of the metabolism to take place. Another category under it, under enzyme function deficiency disorders, is gain of function disorder. This is where an enzyme gains an abnormal function, possibly leading to production of toxic metabolites. In the first one, there is lack of production of that enzyme necessary for that metabolic process. But in the gain of function disorder, the enzyme is there, but however, is not abnormal. As such, leads to production of what is not necessary. So that is why we say enzymopathies are as a result of deficiency or being dysfunctional. Examples of common enzymopathies that's relevant to community health practice is phenylketonuria, which is caused by a deficiency in phenylalanine hydroxylase, which leads to the accumulation of phenylalanine, which can cause intellectual disability if untreated in a person. Another common enzymopathy is galactosemia, which is a disorder resulting from a deficiency in galactose 1-phosphate uridine transferase that prevents the metabolism of galactose, which leads to a toxic buildup of the substance in the blood. Now, let's move into G6PD. Remember, a matured red blood cell must produce energy through adenosine triphosphate, ATP, to maintain a normal internal environment. Red blood cells respire. Remember, they are the one responsible for carrying oxygen around in the body. So for them to do this function, the red blood cell have to produce energy to enable the red blood cell to do all its function. And that energy production is through adenosine triphosphate, ATP, so that the internal normal environment will be maintained in the cell. Secondly, to meet up with the energy requirements to function as red blood cells and to ensure the cell volume, remember that this red blood cell have this utmost function of carrying the oxygen around the body. As such, for him to, like I said earlier, must be able to produce an energy that will enable the red blood cell maintain internal environment, meet up the energy requirements to do this function, while still protecting itself from the challenge of oxidative stress. That is the stress that comes from oxygen reactive species that the red blood cell will encounter while moving in the body. And this oxidative stress can come from oxygen reactive species, so that comes from the drug the person have taken or some infections. So the red blood cell is fixing a lot while it's moving inside the blood vessel. It has a function of providing energy for itself to move, to carry the oxygen around. It also has to protect itself from this challenge of oxidative stress that results from oxygen reaction species, drugs you take, and infections. ATP is generated by glycolysis. That is, breaking down of glucose leads to production of this ATP necessary. Why the hexose monophosphate pathway produces nicotinamide adenine dinucleate phosphate, NADPH, and glutathione to protect against oxidative stress. So, in the red blood cell, like we said earlier, the red blood cell has to produce this energy known as ATP through glycolysis. That's breaking down of glucose. It requires this process to occur in the red blood cell for the red blood cell to have energy. The red blood does that within himself. Why the hexose monophosphate pathway in the same red blood cell produces a nicotinamide adenine dinucleotide phosphate and glutathione to protect the red blood cell from oxidative stress. So these two functions occur in the red blood cell: the production of ATP for energy and the production of NADPH and glutathione to protect this red blood cell against oxidative stress from oxygen-reacting species such as drugs and infections. Once all these functions are occurring, the red blood cell will be able to do its function. But for whatever reason, the ATP production is affected, that's problem. That becomes an enzymopathy because there's an enzyme that's responsible or modulates or catalyze this conversion of glucose into energy. And there's an enzyme responsible for the catalyzing of this hexose monophosphate pathway that leads to production of glutathione and NADPH that protects this red blood cell against oxidative stress. So if there's any that goes wrong within any of this pathway, the red blood cell will have issue. So enzymopathy occurs when there is a defect in any of these enzymes in the red blood cell. However, the severity depends on the step affected in the enzyme process and the presence of alternative pathway. That's what's called alternative pathway. If this pathway has an issue, like you are going through a street and the road has an issue, you can follow another path. That's the same way. If there's an alternative pathway for the body to bypass that enzyme that have issue to still reach at the production of ATP or NADPH, fine. But if there's none, then that enzyme, that red blood cell is already having a serious issue. In general, defects in the hexose monophosphate shunt pathway result in a periodic hemolysis that is precipitated by the episodic oxidative stress, while those in the glycolysis pathways result in shortened red blood cell survival and chronic hemolysis. So, like we explained, for any reason that happens that the ATP is no longer being converted for enzyme-related issues, the red blood cell will die very quick and will hemolyze immediately. The body will hemolyze it immediately. That's killing it. But for any other reason that happens in the glycolysis pathway, the red blood cell survival rate will be shortened. But for anything that happens in the hexose monophosphate shunt pathway, it will result to hemolysis once that red blood cell is exposed to any oxidative stress, such as infection or drugs or any other oxygen-reacting species in the blood. So glucose 6-phosphate dehydrogenase deficiency is a condition that results when the enzyme glucose 6-phosphate dehydrogenase is lacking or having issue in the hexose monophosphate shunt pathway. So one can say that the enzyme glucose 6-phosphate dehydrogenase is important in the hexose monophosphate shunt pathway. Thus, the deficiency So one can say that enzyme glucose 6-phosphate dehydrogenase is important in the hexose monophosphate shunt pathway. Thus, the deficiency of this hormone or this enzyme glucose 6-phosphate dehydrogenase result in the most common human enzymopathy that is affecting about 10% of the world population, known as glucose 6-phosphate dehydrogenase deficiency disease. So this enzyme, glucose 6-phosphate dehydrogenase, is important in the hexose monophosphate shunt pathway that is important for protecting the red blood cell against oxidative stress and oxygen-reactive species. So when it had this effect, therefore, the red blood cell will be exposed to unnecessary stress, and which could lead to intermittent destruction of the red blood cell in the body, which presents with jaundice, anemia, and can be fatal at times. Glucose 6-phosphate dehydrogenase deficiency is a genetic condition that affects red blood cells. In a person with G6PD deficiency, the enzyme G6PD, which is important for protecting red blood cells from damage, is either absent or not working properly. When a person with G6PD deficiency is exposed to one of these stressors, such as the oxygen-reactive species, infection, all of that, their red blood cell can become damaged and break apart, a process known as hemolysis. This can lead to a shortage of red blood cell, causing symptoms like jaundice, that's yellowness of the skin, fatigue, and in severe cases, anemia. Individuals with G6PD deficiency, their body is unable to properly protect red blood cell from oxidative stress. Red blood cells are constantly exposed to stress and need protection from certain substances like infection, some medication, or food like the fava beans. The G6PD enzyme helps maintain the health of red blood cells by preventing damage from oxidative stress. And the G6PD, which is an enzyme that is responsible for the NADPH pathway that leads to the production of glutathione and NADPH, is important to catalyze that process for the production of glutathione. I know that glutathione is a very good substance in the body that protects the cell membrane of red blood cells and other cells. The enzyme G6PD is found on the gene on the X chromosome. As such, the deficiency of this enzyme affects male and rarely affects homozygous female. But it is carried by females. Carrier heterozygous females are usually only affected in the neonatal period. And this condition is common in East and West Africa. And up to 20% of male affected and 4% of female affected. The condition is inherited in an sex-linked recessive pattern, meaning that it primarily affects male, while females can be carriers or affected if they inherit the gene from both parents. You know, since this particular condition is only found on the X, not on the Y, if a male inherits that defect, affected gene on the X, because man has only one X, automatically the man will be affected. But if a female inherits only one X that have that issue, it cannot affect her, but she becomes a carrier. It can only affect her if she inherits both defects from the father and the mother. That's where it can affect her. But for a man, once he inherits a copy of it, you are going to be affected. This condition can lead to a shortage of red blood cell, causing symptoms like jaundice, yellowing of the skin, fatigue, and in severe cases, anemia. The following are drugs that can induce hemolysis in persons with this type of condition. Remember, we're talking about oxygen-reactive species. Analgesics such as aspirin, phenacetin, the antimalarials like primaquine, quinine, chloroquine, and perimentamine. Then antibiotics such as sulfonamides, nitrofurantoin, and ciprofloxacin can also induce this condition. That is why most of these drugs are contraindicated in patients who have G6PD. Other drugs like quinine, probinicides, vitamin K, and dapsone also would lead to hemolysis in this group of people who have the G6PD. Another thing is favism. Favism is a condition triggered by the consumption of the fava beans, also known as the broad beans, in individuals with G6PD. So when these people, group of people who have this condition, eat a fava beans, it would result to this hemolysis. And if acute transfusion is not given, the person might die from this condition. Management. The management of this G6PD only aims to stop the intake of precipitants like drugs or food and treat on any underlying infection that can try to trigger this hemolysis. You know already that this person has deficiency of this enzyme. As such, the NADPH and that of the glutathione that is responsible for protection of the red blood cell against these stressors such as drugs, food, and all of that, and infection are not there. As such, those patients must be supported to avoid those foods, drugs, and infections that can lead to oxidative stress as the red blood cell is carrying out function, carrying the oxygen around the body. If not, the red blood cell will be exposed to oxidative stress and will lead to hemolysis and destruction of the red blood cell, and severe anemia might take place.